Hello, Mr. Allman here, and this is your Allman Cast on chromatography. Chromatography is an analytical tool that's used to be able to separate a mixture of chemicals. There are many types of chromatography, the simplest of which, and the type that you will be performing in class, is called paper chromatography. In paper chromatography, you're going to use a strip of chromatography paper. Paper, being made of cellulose, is polar. Therefore, any molecule that is polar, through the concept of adhesion, will move by capillary action through the paper. In this, you first need to start with a substance that needs to be analyzed. In the chromatography lab you're doing for our botany unit, you're going to be analyzing the variety of pigments that are found in the chloroplasts of cells that are utilized by the plant for photosynthesis. For this, you're going to need a leaf. Any green leaf will do. You want to take that leaf, fold it up, and use the side of a pencil or a coin and rub along the leaf to deposit some of that pigment onto the surface of the paper. You're going to want to do this several times, moving the leaf around to be able to get a new group of cells, and the end result should be that you've got a nice dark green line across the length of your paper. You can do that on both sides, and the darker the line, the better. The darker the line, that means that there are more pigments that have been deposited from the leaf onto your chromatography paper. Then you're going to take your paper and attach it to your official chromatography holding device. So I'll use a piece of tape, I'll attach that on there. Now I'm able to suspend my chromatography paper above a chromatography solution. Into my graduated cylinder, I'll fill this up with chromatography solution up until the point at which the bottom of the paper is just touching the liquid. I do not want my green pigment to be submerged. I want the level of the liquid in my beaker to be below the level of my pigment. That way, what's going to happen is that as the chromatography solution is drawn up by adhesion into the paper, it's going to move higher and higher and higher up the length of that paper. Now the pigments that have been deposited on the chromatography paper will move along with the solvent. But as they move, each pigment, based on the size and polarity of each of those molecules, they're going to move at different speeds. The end result, after you have allowed the solvent to move up 3/4 to most of the way up of that paper, you can then remove it and lay it out to be able to dry. Once it's dry, you want to analyze to see the results. What you're going to see is that as the solvent moves up, at the very top of the paper is going to be the solvent front. The solvent front is going to be the part where you can see that the paper has become damp because the solvent has actually moved up that far on the paper. Then there should be a series of lines, each of which may or may not have a different color to them. And each of those lines represent a different pigment that is found in the leaf. What you then will want to do is to be able to make some measurements using a ruler to figure out the RF value. The RF value is going to tell you the relative affinity that each of those pigments has towards the solvent. The solvent in our case that we're going to use is going to be a combination of acetone and alcohol. So that's a polar molecule. It's not water, but it is a polar molecule. The RF value is going to be calculated by measuring the distance traveled by the substance. So if this red line represents one of my pigments, I would measure from the bottom of the paper up to that red line. Then I would divide that by the distance traveled by the solvent front. So the distance again from the bottom all the way up to that solvent front. Those two numbers are expressed as a ratio, and so the range is going to be anywhere from 0 to 1. If the RF value is 0, that means that the pigment is in the stationary phase. it has no attraction towards that solvent. The solvent passes right over and does not pull any of that pigment with it. If the value is very close to 1, that means that the pigment is at or very close to the solvent front, which means it has a very high affinity towards that solvent. As you look at this, you should be able to see several different colors, in this case representing chlorophyll A, chlorophyll B, the xanthophyll, and carotenoid. You should be able to find at least 4 separate pigments as you analyze this, assuming that you have allowed each of those pigments to travel a far enough distance so that they have time to separate from each other. One word on safety. The chromatography solution is highly volatile. Volatile meaning that it has a very easy for it to vaporize and become a gas. And so you should perform, you can set up your chromatography solution at your lab table, but you should move your chromatography setup to the fume hood and make sure that the fan is on because we don't want these fumes to fill up the room. Finally, because of the danger of the solution, you should also wear goggles during this experiment to be sure that your eyes are protected. Thank you for watching. If you have any questions before we start, please let me know.